Welcome back, fellow mitochondriacs, for another episode of Cancer is a Mitochondrial Metabolic Disease. In particular, this is another episode of the podcast with Professor Dom D'Agostino from the University of South Florida. He is in the Department of Molecular Pharmacology and Physiology. And I stole this from his department bio, and it said his academic philosophy. Just check this out: educating future scientists and clinicians on metabolism based research and therapies to advance the treatment and prevention of disease. I just really love that philosophy that he has there. And in this part of the podcast, we're going to talk about ketones, its effects on cancer, oxidative therapies such as HBOT and IV vitamin C, why oxygen via a mask is not the same as HBOT, and are soft shell chambers or soft shell hyperbaric oxygen chambers useless? What is the best evidence-based protocols for HBOT in this current time? And much, much more. So without further ado, let's get into it. Segue, Doc.
Yeah.
So I think there is a lot of fear within this kind of community. And, Dr. Seyfried's done a lot of work on this, and I'm not sure if there were papers that you were associated with. I mean, they're on ketones, so they may have been, but can can ketones be burned by non-mitochondrial containing cells or cells that don't have functional oxphos? Is there a way that they can be fermented through mitochondrial substrate phosphorylation in any way that you're aware of?
They're not fermentable, but I think ketones can be potentially used as a precursor for the biosynthesis of lipids. So I know that acetoacetate plays a big role in synthesizing myelin. So for neuroregeneration, that's why ketones are quite remarkably effective for traumatic brain injury because they can help repair the myelin sheath. And if you radio label acetoacetate, then a lot of it gets incorporated into a myelin, which is a fat-like substance or fat-containing substance. So, and I think maybe other people have shown too with radio labeling and tracer studies that ketones may not be ketone metabolism for energy is marginalized sufficiently. And I think it's not maybe all or none, but I think generally speaking, ketones require oxidative phosphorylation to be, to generate ATP from that. But I do think they could be directed for biosynthetic processes. And we know that a big part of the Warburg effect, we're talking about metabolism, but a big part of that is to redirect metabolites for the biosynthetic expansion of the tumor biomass.
Yep, 100%.
And that could be achieved in part by ketones could be feeding into that. I mean, you could say that about anything. You could say about anything that you eat,
Anything, truly.
amino acids.
And I think that's where what happens is that these papers, they'll say, oh, this cancer is using beta hydroxybutyrate, acetoacetate, whatever, to grow, right? But what they're missing is that those biosynthetic pathways that's being used as is ATP dependent. So therefore, if you give a cancer probably, I could be wrong here, but I've seen the papers that I've seen is it looks like if you give a cancer that has unlimited glucose and glutamine, beta hydroxybutyrate, fat, whatever, amino acids, of course it's going to be used for biosynthetic and it's going to probably grow better, whatever. But if you were to restrict the ability for those pathways to be active, if they're ATP dependent, then it would make sense that that would not be it would be a non-issue essentially at that point.
Mhm. Yeah, I mean, we've published, and Tom Seyfried is on the paper. I think the title of the paper, it's International Journal of Cancer, a good journal, and it said ketone supplementation, extends survival and suppresses, tumor growth or something like that. It's International Journal of Cancer, where we actually used 1,3-butanediol and we used a ketone ester, and that worked in that way. Probably in hindsight, we would use pivot to use some of the other compounds, but we also took the ketone salts, the DL ketone salts, and we grew the cells in culture and we actually grew them in 25 millimolar of glucose because that's how you actually
Wow.
expand the. So that's actually the standard protocol that Tom Seifert sent us to grow the cancer cells, and it just makes them like super highly aggressive. So in the presence of 25 millimolar of glucose, we added the ketones, and the ketones decreased cell growth and decreased proliferation, and also increased dead cells. So we did Ethidium homodimer 1 and calcium to look at a live dead cell analysis, like a standard kind of assay for that. So we saw suppression of tumor growth, of cancer cell growth, suppression, and we also saw greater cell death by not changing anything, the glucose in the media, not changing glucose or glutamine, but simply adding ketones. And that study was done by Dr. Angela Poff, and that was part of her PhD dissertation. Prior to her doing those studies, I had a medical student do that. And the title of her poster for her student presentation was that ketones kill cancer. And I remember a lot of students were going, and I was like, okay, the results seem sometimes like students are, could be basically picking cells out. So when Angela came into the lab, her thing was to replicate that finding. And I think we might have used a couple of different there were brain cancer cell lines, but she replicated it, and I think others have. Actually, there was a paper by Skinner, and he was at University of Florida, and I tried to get in touch with him because he used a neuroblastoma cell line, threw on ketones in a dish, and saw more cell death and less proliferation. And then Eugene Fine from Einstein did it with acetoacetate. So and then my student did it, and it was like, wow, these seem too good to be true. And then Angela replicated, and then we ended up publishing that.
Wow, that's
Yeah.
incredible
And
because that's a
that's not
lot of
changing
glucose.
anything.
Wow. I mean, I can't even
Yeah.
imagine the milligrams per deciliter that would equivalent to, to be in a human patient, but that's an incredible amount. And you're saying that you didn't have any restriction of glutamine either, and still,
Nope.
wow.
Yeah, we use like a serum and that has like glutamine in it, and maybe we supplement it a little bit, and we use like DM, EM. We have like a whole cocktail, and we grow the VMM3 cells with like, a solution that basically makes them grow as fast as possible. So the idea is make them grow as fast as possible so we can get enough cells, and then we collect them, and then we inject them into the flank of the tumor, and then it's syngenic model, so they just go. The cancer cells go to the liver typically. They pepper the liver with all these micrometastases. It's really gnarly, disgusting look. It's like oozing tumors coming out of the liver. And then the cancer cells also, they go to the kidney, they go to the lungs, and then after about a week to 2 weeks, they get into the brain and they start causing uh, brain tumor. So that is what's kind of unique and controversial about this model is that you can inject it into the flank and then it actually causes, brain cancer. So most glioblastoma cells and cell lines, it's very controversial, but most people who study glioblastoma kind of say that, a glioblastoma should not grow outside of the brain. And that's what's kind of unique about this tumor. And Tom would argue that. And he like, I had some pushback about this model. And he was like, it's turned on. I mean, there are, I guess, reports where someone had maybe gotten a liver or something from a brain tumor patient, and then that liver had like the glioblastoma cells. So there is evidence of glioblastoma cells going and causing metastasis to organs of the body. And
Yeah. I
it's
can't say I've
maybe
ever seen
not,
it in real life. I can't say I've seen a GBM patient ever have Mets anywhere else, but
yeah, Tom Seifert is sending you a couple papers that, will convince you that it's at least possible. But I do think that there's so many mutations in these that they're just so glycolytic. And they also have like a phenotype, like they have a unique phenotype. They're like a tumor associated macrophage
hmm.
kind of, and they're a bit different than they're like this transformed glioblastoma cell that's like a tumor associated macrophage hybrid kind of thing. But I do think that it's that model is one of the best models
Awesome.
out there, and they're kind of unique to study because I also think they suck up glucose and glutamine, and I think that's relevant to this discussion and also highly relevant to creating therapies to manage
Yeah.
this disorder, to
Well, do you mind
understand.
if I switch gears just because I know you have also tons of expertise with oxidative therapies also. And I
Mhm.
just wanted to, one of the things that I asked Dr. Siegfried, and I don't know if I got the answer I was looking for, but in 2017 with his press pulse paper, which I believe you were on that paper,
Yeah, the senior author. Yeah, Thomas. Yep.
and And then in 2024, December, when you guys published the new one with the clinical
Yep.
ketogenic,
Mhm.
that one, I think Duraj was the first author on that one,
Yeah, Tomas.
but, but there was no oxidative therapies mentioned in that second paper. And I was like, do you believe that,
There was hyperbaric was mentioned in
but
there.
it wasn't
It
like on
wasn't
the,
super prominent. It wasn't on the figure, maybe.
yeah,
Yeah,
it was on the
because
figure.
I wrote the section.
It wasn't super,
Yeah.
yeah, it wasn't super prominent in the article. And I just wondered, like, did something happen between 2017 and 2024 that made you guys believe that oxidative therapies are not required for press pulse, or do you think that it was just something else?
No, I mean, in hindsight, I should have spent more time expanding that, oxidative stress. But I think the important take-home message was that ketone metabolic therapy is putting the patient into a glucose ketone index of 1 to 2. I push back a little bit and say 1 to 4. And it de-emphasized anything that was not uniquely metabolic. So it de-emphasizes, and hence the title of the paper, right? The framework for ketone metabolic therapy for
Right.
glioblastoma. So I think maybe I wanted to get more of the hyperbaric in. Also, the hyperbaric, we don't know the dosage, the timing, and then the scheduling of that. Also, I have to say, as someone who studied oxygen toxicity seizures, is that when you have a glioblastoma, I am hesitant to tell people to jump into a hyperbaric chamber at the standard hard shell chamber, shall we say, dosages of 2.5 atmospheres or 3 atmospheres, which we have published on, right? Because having a tumor makes you much more vulnerable to having a seizure that could be evoked by oxygen tox. So my recommendation is to do like, low dose oxygen, like 1.3, 1.5, do that for a couple weeks. And you have to understand that that's more like the bleeding edge of science. So this idea of creating, to metabolically compromise the tumor and then hit it with an oxidative stress. So I'm almost more likely to recommend something like metformin or recommend some other drugs like IV vitamin C, right? So you give millimolar concentrations of circulating vitamin C, functions as a glucose antagonist. Ascorbic acid uses the same transporter as glucose, so it's functioning that way. And also, the high dose of vitamin C functions as a pro-oxidant. It drives something called the Fenton reaction, and that you could actually enhance iron-induced oxidative stress. And we talked about the porphyrin overdrive
Hmm.
hypothesis being very prominent, especially in GBM. So driving that. So I'm a little bit more comfortable mentioning that. And with the hyperbarics, I think there's a lot of potential for hyperbarics, but I think not everybody has access to that too. So, and that becomes, an accessibility problem for patients that
Huge,
just
huge
do not have
accessibility problem.
access
At
to
least the hard
hyperbarics.
shells, at least. I mean, a lot of people can, even
Yeah.
in rural areas, can have access a lot of times to the soft shells, which kind of leads me to
Yeah.
some of the questions I have later about like that. But do you think that HBOT is the same? It's better, worse than other oxidative therapies, potential therapies? I know you mentioned IV vitamin C. There's other people who talk about ozone or photodynamic therapy. Do you think that one has an instinctive, is it one better than the other in your eyes?
Well, I gotta say, I mean, like just north of me is the Aviv Clinic, and they have like literally tens of thousands of, older patients that they do hyperbaric oxygen therapy for longevity. And they have greater, cognitive function. They do better reaction times, decision making. They're using it as a cognitive enhancer. It stimulates the production of stem cells and the mobilization of those stem cells to hone in on sites of injury and inflammation. It has a lot of remarkable property. The folks in Israel showed it lengthens telomeres, so I'm a little bit less convinced of that. Although, or if that's beneficial, not. I believe in the results, but maybe the interpretation of the results. So, and I'm in touch with, NFL guys, sports teams, and people. So hyperbaric oxygen, what it does, it enhances wound healing. Like everybody, I mean, it's FDA approved for that. So when you're banged up on the football field or you're like a special operator, you're just like really going at it in the gym, right? And you get inside a hyperbaric chamber, it's really augmenting those healing processes. And it's creating oxidative stress, but the adaptive response to the oxidative stimulus and the repair mechanisms are what happens,
Right.
right? So, and I think that could be different in older patients because their adaptive response to an oxidative stress stimulant can be different. So I know, I mean, I'm in my 50s now, so when I go and I do like a super brutal squat workout or deadlift workout, it takes me like 5 days to recover now because I don't have like, the testosterone and all the hormone stuff that I did when I was 20, right? And I recovered like in a day or two. But now I kind of liken it to that, right? So if you go and do a super hard workout and then take blood work and look at that blood work and say, well, whatever you did, stop doing that because you have all these inflammatory markers, oxidative stress. The same thing kind of happens with hyperbaric oxygen therapy, but it's the training stimulus and the adaptive response to that stimulus that produces the beneficial effects. So I'm a firm believer that if you're in a state of therapeutic ketosis and you do hyperbaric oxygen therapy, the adaptive therapeutic beneficial effects of that hyperbaric therapy will be greatly augmented. And you'll also decrease the potential for oxygen toxicity. I mean, that's clear, the anti-seizure effect. And it negatively impacts the cancer cells and positively impacts the healthy cells.
Got it. And do you think, I mean, is there benefit to being in HBOT without being in ketosis or without, addressing glutaminolysis, or is it a waste of your time and money, like type of a thing, or is there benefit on its own?
No, like all of our literature is actually without ketogenic, all of our literature that's not from my lab, right? So we're the ones that, but what I can say, there's 14 different FDA approved applications. So one is radiation necrosis for cancer patients that have, damage from radiation therapy. And it was observed in attending the Understanding Hyperbaric Medicine Society meetings, like they would show these case reports and like, oh, the patient like healed up and somehow the tumor went into remission and the patient said, so, but they were talking about it, by the way, like here's what hyperbaric oxygen did to reverse this radiation necrosis and enhance, and this cancer patient just seemed to be like, it was one after another, like these patients were doing better. When we do hyperbaric oxygen therapy with the standard diet, we did not see an increase in survival. There was like a little bit of a trend, but it didn't improve survival statistically significant. If we did a cohort of 50, maybe we would have had statistical significance. But what it did do, it enhanced the tumor suppression effect of the ketogenic diet. So there's some unique synergy there. And I think, that if you have cancer and you're undergoing radiation therapy, insurance will cover it. But the more you improve your metabolic health, I could say if your glucose is high and and you don't have good metabolic health, I worry about the oxygen somehow augmenting maybe, inflammatory processes that could, worsen the situation. But I think each case needs to be, I mean, each individual is like a unique metabolic entity. And I think hyperbaric oxygen needs to be titrated to the individual. And what we don't have are biomarkers that we can look at as people are going through hyperbaric therapy to titrate the dosage, timing, and scheduling of that hyperbaric therapy.
Yeah.
So this is like, what we always discuss when we get together as hyperbaric, experts in the field. Like, we need to, and there's a couple biomarkers that the best one we have now is HSCRP. So if you have a patient and their HSCRP keeps climbing with hyperbaric oxygen therapy, which I've seen on the 5 day a week schedule, back that off to 3 days a week, Monday, Wednesday, Friday. And then you often see, they continue to get benefits, but the HSCRP goes down. That means the oxidative stress load over time,
Interesting.
they're not adapting to that. So
What a, is there help me understand the benefit of HBOT as an oxidative stressor for cancer compared to just, I don't know, taking like a non-rebreather mask and just breathing 100% FiO2. Like what additional benefit is there? Because I've had people ask me about that and I'm like, I don't think it's going to be the same. But I just wanted someone like you to help me understand that better.
yeah, that's a big question. And I think it caused a lot of confusion, right? Because people just breathe 100% oxygen, right? You're going to Interestingly, and I've done the experiments back when we used to do like larger animal experiments. If you measure, I'll start off by saying this. If you do an experiment with an animal model experiment, or just take a human, and you put an oxygen electrode in the human's brain, and you measure the partial pressure of oxygen in the brain tissue, breathing normal air, and then you put a mask on them, which is 100% oxygen, what do you think would happen to brain PO2?
It's going to go
Question.
through the roof, I would imagine.
No, it goes down. It goes down. And it goes down for a number of reasons. So when you breathe 100% oxygen, it causes hyperoxic vasoconstriction. So unless you titrate in CO2, and that's called carbogen, right? So that's another thing that we could talk about. Yeah, breathing 100% oxygen actually causes what's known as hyperoxic vasoconstriction. And this, I was not fully aware of that until we were doing the experiments, and I was like, "This can't be right. Let's switch the oxygen electrode." And then I was like, "Oh, okay, yeah." So, and it's in the books actually, like in old, like physiology books, maybe
Uh-huh.
not like in medical books. So, under normal conditions, a normal healthy person's hemoglobin is saturated, right? And hemoglobin is how we get oxygen into the cells. When you breathe 100% oxygen, hemoglobin's already pretty much
Saturated.
saturated. If you're on the sidelines of a football field or you have a patient with COPD and you throw 100% oxygen, you might jump it up a few percentages, so you might increase it a little bit. But for all intensive purposes, hemoglobin's saturated and you're not going to get more oxygen into into the tissues, right? You may actually decrease it through vasoconstriction. So what you have to do is you actually have to increase the partial pressure of oxygen. The dose of oxygen is a function of the concentration of oxygen times the atmospheric the pressure, the barometric pressure. So that will dictate the partial pressure of oxygen in the tissues and in the blood. So by increasing the barometric pressure, you're pushing more oxygen into the liquid phase of blood, and that's called the plasma. If oxygen is at a higher concentration in the plasma phase, then that gets into all the nooks and crannies of a tumor that has erratic, blood vessel growth, right? So a tumor, the core of the tumor becomes hypoxic because the red blood cells kind of get trapped in like the erratic blood vessels and things like that. Hyperbaric oxygen, by virtue of pushing the oxygen into the liquid medium, the plasma, rapidly reverses tumor hypoxia and super oxygenates the tumor by getting the oxygen into the actual liquid phase, right? And when you do that, the mitochondria are all screwed up and damaged, and when those mitochondria, their tissue PO2 goes from 3 millimeters of mercury to 2000 millimeters of mercury. From that causes a massive oxidative burst that's kind of site specific, right? Like the healthy cells can handle that, but the tumor cells are used to thriving in a low oxygen environment, pumping out lots of lactate that like protects them from the immune system and things like that. So you reverse the tumor hypoxia, you hyperoxygenate the tumor, and then that tumor actually starts to die from the high concentration of oxygen making reactive oxygen species. But if you were to then hit that tumor with radiation, then boom, like actually the radiation, when you hit the tumor with radiation, it kills the tumor by generating reactive oxygen species,
Right.
which is entirely dependent upon the PO2 of the tumor.
That's what you were mentioning in the very
So,
beginning. You said it's dependent on the concentration of the PO2 in the tumor, you're saying, right?
yeah, yep. So there are a lot of clever ways that you could reverse tumor hypoxia, hyperoxygenate, and use a toolbox of different drugs to increase that oxidative stress 5 to 10, or maybe even 100 times higher, especially if you knock out like a few key enzymes with relatively non-toxic compounds. And that's the direction I want to go into.
There was a paper that was published. It
Sure.
was pretty cool. It was a paper that was looking at Aristin, and Aristin blocks SXC, the SLC7811, which is the
Mhm.
cysteine glutamine antiporter.
Yep.
And they were showing that it was, I think it was a breast cancer or maybe a ovarian cancer model. And they gave they gave Aristin at concentrations. They looked at the viability of the cancer cells, and it was nothing, right? And then what they did was they added iron by itself, and it had an effect. And then when they added them both, because the aristin is inhibiting glutathione production through the lack of cysteine that's getting into the cells, they added iron. It was like 99% that were gone. What about iron? What do you think iron could also be used
No,
therapeutically like that?
yeah, so that's the idea. So that actually you can kill cancer cells via iron by vitamin C. So the pro-oxidant effect of vitamin C drives that, but also the 5-amino levulonic acid, which is the precursor to porphyrin, right? And that porphyrin sucks up the iron. It brings, and the GBM cells suck that up, as do every other cancer cell that I can think of that I know of. GBM and pancreatic cancer are just much, they do it a lot more. They have this porphyrin overdrive. So that's essentially what you could do by using a relatively non-toxic compound, have the patient consume it, and then that site specifically jacks up iron by several fold higher. And then in the context of reversing tumor hypoxia and hyperoxygenating the tumor, then that creates more oxidative stress. The tumor cells will try to defend itself through a number of different ways, pentose phosphate pathway, but also heme oxygenase. And there's probably about a half dozen different enzymes, and a few of the key ones can be knocked out with different compounds. One of the compound I spent my whole PhD like studying. I was studying something different, but that could be repurposed for this. It's actually used for hyperbilirubinemia, and it's 10-protoporphyrin 9. It could be used to you could leverage that potentially to knock that out. And we have a lot of ideas that I think could make hyperbaric oxygen in the context of a few off-the-shelf repurposed drugs far more lethal to cancer cells than radiation therapy, and essentially non-toxic.
I mean, even
In
aspirin
theory.
and metformin block the SLC7A11. I mean, so does sulfasalazine has an effect there too. So like
Mhm.
there's several things that are already there that have probably
Yeah.
good pharmacokinetics, excuse me,
Mhm.
that maybe could be able to augment that as well.
Yeah, and those things are, relatively safe. Everything's going to have off-target effects, but you really want, there are ways that you can, combine these things in ways that are more than additive, that are synergistic. And that's what we're kind of shooting for here. It would be, we envision a cancer therapy where like patients undergo the cancer therapy and they come out in more robust health than they went in. So
Right.
now, I mean, you just like IV poison and radiation, you come out like with a lot of other problems too if you survive the cancer. But
To mention
we
secondary
envision,
malignancies. I mean,
yeah, we envision, and central to that too is going to be, and probably should talk about it more, just like exercise. Exercise oncology needs to be like every cancer department should have exercise oncology. They should have metabolic oncology, exercise oncology. Uh, we need to leverage, light therapy and just movement. Movement is medicine, and it doesn't take much either, too, to do this to mitigate also cancer cachexia. So we've had students in my lab focus exclusively on mitigating the muscle wasting associated with that. And cancers have a remarkable anti-catabolic effect. That's really their evolutionary function. Is that if we stop eating and we quickly start metabolizing fatty acids for fuel. And if we didn't make ketones, we would quickly catabolize the gluconeogenic amino acids in skeletal muscle and in our organs, and we quickly waste away and die. So then, because we generate ketones, those ketones, by virtue of supplying energy to the central nervous system, become incredibly anti-catabolic.
hmm
At first, I was just thinking, oh, it's just an energy, just because they supply energy. But no, they have uniquely anti-catabolic mechanisms through signaling pathways. So we've published articles. Dr. Andrew Kutnick, who did his PhD in my lab, published a couple articles on the anti-catabolic effects, and we've published that in the Journal of Cachexia and Sarcopenia, a good journal, and just show we study cancer cachexia, but we also have a model where we inject with lipopolysaccharide, LPS, to create inflammation, and
hmm
that creates muscle wasting and anorexia. We could largely reverse that too with ketone supplementation. So
Fascinating.
independent of dietary intervention, feeding ketones reduces the inflammatory cascade and also mitigates much of the muscle wasting associated with cancer cachexia and also LPS-induced muscle wasting and inflammation.
Yeah, I read a paper recently. It was talking about the, dysbiosis and how that LPS that probably gets through
Mhm.
via, intestinal permeability leads to cachexia.
Yep.
And it's just fascinating. There's multiple mechanisms. Lactate also kind of like leads to like thermogenesis and all this other stuff. It was a lot of different mechanisms, but it seems like it could be pretty much solved via a ketogenic diet and exercise. It seems like that's like the main thing that seems to be helpful. I mean, to help reverse the
Yeah.
kind of vicious cycles that are happening through the Cori cycle and all that stuff that kind of re-goes back and feeds that. And I didn't realize, I guess I didn't think about this, but like from an energetic perspective, also, given that gluconeogenesis is an energy, requiring, ATP consuming thing, you basically are stealing Peter to pay Paul. Basically, you're giving that glucose back to the tumor. It's going to use it for its bioenergetic stuff, and then it's going to have to steal from
Mhm.
someplace else in the body too. Not to mention the muscle cells. I could probably ask you about 1,000 different things, but I think that one thing I definitely wanted to ask you was, can you give me your best understanding of what a potential protocol would look like for a patient if they were going to employ and it was right for them, hyperbaric oxygen therapy in combination with ketogenic metabolic therapy? I know you mentioned that maybe 5 days on can be a little rough for people because of the excess oxidative stress. Is there any like dosing, timing, and scheduling that is the best of the best literature we have today, right now?
Yeah, there's a paucity of information like in the literature on hyperbaric oxygen for cancer. A lot of it's actually in this standard of care for, you'll find it for radiation necrosis, where like case report, case series, where the patients do remarkably better. I would steer people towards the clinical framework for ketogenic metabolic therapy in glioblastoma. So Dr. Durai, Tomas. So that's a good paper. So the idea then, like just from a 30,000 foot perspective, is to achieve and maintain a glucose ketone index between 1 and 4. And my feeling is that if you can maintain 4, you're doing well. If you can bring that down to 2, especially if you're going to couple that with hyperbaric oxygen therapy, that's going to be beneficial. So that's the big lever. So achieve and maintain that. And then with hyperbaric oxygen therapy, I have not seen any data to indicate that a protocol is better than what we use, which is 3 times per week. We did like Monday, Wednesday, Friday, 2.5 atmospheres for 60 to 90 minutes, given 3 times per week. And I do like that lesser protocol instead of 5 days per week, which is a standard protocol. I like the 3 days per week for the cancer patient because they're already kind of compromised, and I think they need that gap day, and that relative kind of shift can be really, and also logistically, it's 2 less days you have to get inside the chamber, and I think it could be beneficial if, for example, you're paying for like 40 dives or something like that. So I haven't seen anything to convince me that, every other day. I have seen evidence to convince me that doing it every day starts to push some markers in the wrong direction. And then like people are accumulating greater oxidative stress over time, and that could impair, the health of the patient and maybe set them up for some other problems. So doing that, and then, the achieving and maintaining a glucose ketone index would be with a modified ketogenic diet. I'm of the opinion about 20 to 25% protein is going to be favorable for cancer patients to prevent muscle wasting and to get as much MCT as possible that you can incorporate. And the MCT generally is well tolerated if you take it in with the diet. And then, I mean, nowadays, like even with the epilepsy world, like you have to take electrolytes. Electrolytes are really important because there's a diuretic and a natriuretic effect. Natriuretic meaning that you lose
Oh,
sodium on a ketogenic diet. So like Keto Start, which is like a sodium beta hydroxybutyrate, it's got potassium, magnesium, and calcium too. So to take an electrolyte ketone, a ketone electrolyte supplement is going to be important with the MCT, and then add that more or less to knock down your GKI if needed. and it's not, they're way cheaper than like an ester or something like that, and they taste good too. So that's a big kind of
Would you
why.
take that? Would you take that before getting into a chamber? Would you take an extra ketone supplement before you get into a chamber? Not only maybe to make you not feel as, I don't know, hypoglycemic or whatnot, but also because of a maybe a clinical benefit that you would get from having a higher BHB
Yeah.
level.
I mean, we're of the opinion everything that we've done shows greater efficacy and safety for sure. I mean, my whole career is built on ketogenic mitigation of oxygen toxicity. So I can, firmly say that it's going to make a safer dive. And then those ketones too, in the context of, so the ketones have a vasodilation effect. So by dilating, especially the LBHB, augmenting cardiac output by decreasing peripheral vascular resistance, you're going to basically push more oxygenated blood into the tumor, right? You're going to get better circulation. So if you acutely take something that's going to then increase circulation and also have like epigenetic effects and other things that's going to be beneficial. And in addition to it's sort of protecting the brain from having a seizure too. So I mean, in theory, you could also push the levels higher of oxygen, like, but that's, there are experiments that I want to do. I want to actually do ketogenic metabolic therapy, with cancer, but actually do it in an experimental paradigm where I go like twice as high than the upper limit of hyperbaric oxygen therapy. So if we could really blast the tumors in that context and use some other drugs, that would, so it's more like a proof of concept kind of thing.
Mhm.
But if we could decrease the potential for oxygen toxicity by like 500%, and protect the brain from having a seizure, you could theoretically do that. The major implications would be like carbon monoxide poisoning
All
or
right.
decompression sickness. So you push people
And you
more.
just and just to be clear, you just want extra ROS. Is that what you're looking for at a higher pressure like that? Would
Yeah. Oh, yeah.
stacking like IV vitamin C in addition to HBOT be a way to do that without having to go to 5 atmosphere?
Yeah, you could do that. I've done IV vitamin C, and I don't know if you've done it too, right? If you get like,
I've done it personally, not high dose, but I've done it personally. Yeah,
so when you get IV vitamin C and you get like, a liter or whatever, of that, then you have to pee, you probably you got to run to the bath, you got to pee. So like that has come up, I've experienced that and I was like, oh, this would kind of make it kind of tricky logistically if you're going to do this and then have the patient go to the chamber. So I'm of the opinion that could be done, or maybe you could just use less liquid or whatever. But yeah, IV vitamin C could be done before or after hyperbaric oxygen therapy. Because when you give hyperbaric oxygen therapy, that oxidative stress, the oxygen of the tumor comes down within about an hour to one study showed 2 hours. But the oxidative stress is actually elevated for quite some time.
interesting.
So if you do hyperbaric oxygen therapy, then on the same day in the same center, if you can just walk over and then do your IV vitamin C, you would be kind of taking advantage of that oxidative hit and maybe even extending that oxidative stress to the tumor with the IV vitamin C hit.
I'm not saying we're going this direction, but I just think about like, in medicine, if we got to have someone who can't urinate, for a surgery, we just put a Foley in. We do something like that, that we would
Yeah.
able to uh
Yeah.
handle
I mean,
that,
that's a
with
small thing, but I mean, it's something
right.
that I kind of
No, but
experienced.
it came up
Yeah.
today actually.
But they're
Logistically,
little
we can't
things.
get
Yeah,
it.
they're
I've
little
heard
things
that.
that kind
So
of
yeah,
come
I
up.
get
Mhm.
that. Do you think there's any other ways to enhance like the day of HBOT, like other, I don't know, drug supplements that you have experimented with that could enhance it? I mean, Dawn before, like, or what
Mhm.
other else you're talking about? Sodium phenylbutyrate, I've never used clinically ever, but I've heard of it. And I know my understanding is it scavenges glutamine from the blood. They use it for like what, inborn errors of metabolism or something like that. But
Mhm.
like, are you guys using that like before a dive somewhere?
You could theoretically do that. We've had some mixed results. It doesn't like increase cancer, but like less than optimal results. So I'm hesitant to like mention anything that doesn't have like a robust effect,
Got
right? So I'm looking beyond that. And also I look feasibility and safety. So one thing that you may want to do is you do metformin before and maybe take upwards of like, 2000 milligrams, maybe even 3000 milligrams if you could tolerate that before. could be beneficial. Achieving a lowest GKI is possible, could be beneficial. And maybe in the context of glutamine, EGCG, so really strong, like an EGCG extract, could be beneficial. There's good data for that. Um, and yeah, and then sodium phenylbutyrate could also be used and combined with that.
I've seen it. It's expensive. I mean, it's
Yeah, and
unbelievably
that's why I
expensive. So that's why I think
hesitate.
it's kind of like,
It
yeah.
used to be really cheap, but there was like this ALS study that was done that used it and everybody wanted to get it. And then we were getting it from a supplier, and then it's like ten times higher. It's like price spiking. It's unfortunate. Um, but yeah, there's different ways. I mean, you can probably source it from China and get it pretty cheap. So, I mean, it's like one of those things that gives you, but DCA is another one,
Yeah, DCA.
dichloroacetate, a very simple molecule. We've created some DCA esters too, but we've never tested those. But yeah, DCA, metformin, hyperbaric oxygen, GKI of 1 to 2. Like in theory, I'm not giving medical advice,
No,
but
I
I'm just,
understand.
promoting things
Would you
or
see it
suggesting
be, what would you
theoretical.
see the length of the need for therapy for HBOT? Is it like 2 weeks? Is it 6 weeks? Is it 3 months? Is it a year? Like, do you have any idea like what a realistic time frame would be to see it have an effect on patients? Because it's also expensive, very expensive.
Yeah. Well, I've been reviewing a lot of data that's using it for TBI that occurred like in early childhood, and anywhere from 40 to 60 dives always produces beneficial effects. So maybe like the patient could be thinking about it, hey, to reverse fatigue from cancer and things like that. So you could think about it as like not only hitting the cancer, but
Right.
increasing, your vitality. And everything that I've seen, typically involves like, 20 sessions minimum to 40, and then some studies, 60 sessions and that 3 days per week. But my thing is that, if you're doing it and you're getting benefits from it and you're continuing to get benefits from it, then maybe don't stop. Another thought is just 3 weeks on, 3 weeks off. So, three three dives per week for 3 weeks. It's like 9 dives or 9 or 10 dives. Take 3 weeks off and do it again. Could be. And a lot of, some people find it hard to do the ketogenic diet. So maybe they could just do time restricted feeding, intermittent fasting. One of our keynote speakers was Dr. Walter Longo. He's doing
Hmm.
work on the fasting mimicking diet.
Yep.
So I often have patients that reach out to me that are vegetarian or vegan. And sometimes I steer them in that direction, and feedback from, well, he presented some very compelling results at our metabolic health summit. I think he has a glioblastoma study ongoing now on the fasting mimicking diet. And I know some people at Moffitt are using it too. My feeling is that a restricted ketogenic diet could be done 5 days per month. And that's like a fasting mimicking diet too. If you kind of, cap the calories at 600 calories or
Hmm.
whatever. So you could conceivably do that, but there's calorie restriction, time restricted feeding, restricted ketogenic diet, a ketogenic diet ad libitum. I would not encourage people to go down that path of restriction if their BMI is like under 19 and if they don't have weight to lose. I would be hesitant to encourage them to restrict
Is that just
calories.
hitting would it just be like at maintenance for those people or even like slightly above maintenance? Is there any harmful effects of being above maintenance?
Well, I would do, yeah, maintenance, right? Because you don't want to lose muscle mass. And maybe prior to the dive, going into the dive, you want to do, 18 hours of fasting before getting in, and then, just get back to the low carb diet after. So you might want to, do like at least 12 hours of fasting, 1 to 2 days of, 12 to 24 hours of fasting before doing the dive, and maybe taking some ketone supplementation to just achieve and maintain that glucose ketone index would be good.
Dom,
Yeah, I mean,
I know you get a hard stop at 3, and I want to respect that. But I have one question that I think is going to affect a lot of people, and that is, are people who are in more rural areas or don't have access to these hard shells, are they just like SOL, or do these soft chambers have a role as well? And what would you do to a protocol to make them as effective or almost as effective as a hard shell? Or is there a way to do it?
yeah, I get that question a lot. And actually, my original recommendation kind of is that The data that we have is on 2.5 atmospheres, and that requires a hard shell chamber. But we were just going for the maximum effect, right? We did do studies with 1.5, but it was a small cohort. I think it was 6 to 8 animals, and we just saw a trend, but we didn't see increased survival, statistically speaking. So we can't say, but if you look at the data, there's a trend, like it didn't increase it, right? With lower doses, and that's that's an animal model, so it may not replicate.
Was that an animal model that was restricted of glucose and glutamine, or was that just an animal model on HBOT at a lower dose?
We did that standard diet and also with a ketogenic diet, and we did not see statistically significant increase.
Okay.
However, there's a group, I think, in Germany and maybe one other group that used 1.5, and I think, and it was a different model, and they had like a consolidated tumor, and they saw effects. So, but my recommendation is to cautiously even start out, even if you have access to like a heart chamber, to start out at like 1.3, 1.5, especially with like a brain tumor or something like that. You don't definitely don't want to push the limits, and work with your oncologist, be transparent about what you're doing and start out at that. And we know that if you're increasing the barometric pressure, right, if you're not just breathing 100% oxygen at 1 atmosphere, but if you're increasing that barometric pressure, even a quarter of an atmosphere in like an off, a soft chamber, which is that, the clinic right across the street from me that treats people for different things. That you're getting more oxygen into the plasma and you're reversing tumor hypoxia. So that could be beneficial. And it's feasible and it's cost feasible too. I think, when one place here charges like $30, another charges like $50 per session, many pay out of pocket. So but that's relatively, cheap for some people to handle. Not like, it's a cost burden, but I think I heard through the grapevine that the new administration is going to prioritize hyperbaric oxygen and even expand the standard of care, or the applications that this could be used for. So,
Be
so that
awesome.
was good to hear.
That would be huge if insurance
Mhm.
could cover it. That'd be amazing.
Yeah.
All right. Well, sir, thank you very much. I appreciate it. I'm like I said, I probably have 1,000 other questions to talk about DCA or other compounds
Yeah.
that I would love to pick
Do
your brain
part
about
two. Yeah.
or two. But
Yeah.
thank you very much. Appreciate it. And until next time.
Thanks for having me, Casey.
Yes,
Appreciate
sir.
it.
Thank you. Bye bye. Wow, what an action-packed podcast and discussion that we were able to have together. I learned so much. There were so many different ideas that were teased in this podcast and kind of new directions that has led me down to look at research, especially regarding the porphyrin overdrive, the 5-ALA, not only as it's just a molecule, but as something that could be potentially leveraged for photodynamic therapy. Regardless, I'm probably have to watch this several times over and mull over some of the points that were made here. If you like videos like this, if you like podcasts like this, where we have guests on who are experts in their respective fields who can help move this movement forward, please like, share, and subscribe. And until next time.